Daniella Saetta, who is from also the United States, Arizona State University. She's a PhD candidate there, and she has been an enthusiastic urine diversion researcher since 2014, and a urine summit attendee as well. So this presentation is Bacterial Communities in Urine Collected Solely with Non-Water Urinals. Was fecal contamination avoided? And please tell us, Daniella.
Hi, everybody. My name is Daniella Saetta, and I'm a PhD candidate at Arizona State University, working with Trevor Boyer. Okay, and so the title of my talk is Bacterial Communities in Urine Collected Solely with Non-Water Urinals, and Was Fecal Contamination Avoided? So we've talked about this all day today. I don't want to spend too much time on this slide, but thanks to Nancy, she gave us the big overview of the motivation behind urine diversion. And Randall or Dylan earlier today gave us an overview of the urea hydrolysis process, where urea is hydrolyzed into ammonia and bicarbonate due to the presence of the urease enzyme. And so as this happens, the pH of the urine increases from 6 to 9. The conductivity increases because you're adding charged molecules into solution. Ammonia increases because it's a direct product of the reaction. And phosphate, calcium, magnesium precipitate because you're increasing the pH to 9, where they become supersaturated and precipitate out, which could be a problem for our fixtures, leads to clogging. And so in our group, we do the opposite of what Dylan Randall has talked about. We add acetic acid to inhibit the urease enzyme. And so bacterial communities. So the general consensus in the literature is that fecal contamination is introduced from the use of urine diverting toilets, where there's an interface between the back bowl and the front urine collecting bowl, and fecal material can enter the front urine collection bowl as it exits the body and as it is being collected. And it has implications in terms of storage time needed to inactivate bacteria, the treatment technologies used, and then is it in the fertilizer products that we're producing? And so in our research, we were thinking maybe we can avoid fecal contamination by using urinals alone to collect urine. And so we'll talk about that today. And so for this research, I created a Urinal-on-wheels, which was this big box that allowed us to study our systems with real urine. And so basically we have this big box. Urine is collected into this big tank. We have sensors and pumps to add the acid at the urinal, so the acid comes through the front. And so we have pH, conductivity, temperature inside the tank in real time. And then to compare this real world system, we made a glass urinal in the lab, who actually one of the attendees today has seen in real life. And so we would add urine twice a day to this urinal, and we collected the samples from inside the tank. So this is the tank and this is the trap. So these are some of the results that we got from the study. I'm not going to go into this today, but this study has been published, and so I'll invite you to look into it. This is the citation for the study, but basically the sensors that we had in the tank could measure in real time, and so we got some really cool graphs. And then I took samples on a daily basis to characterize the chemistry inside the tank. And so when we look at bacterial communities, there's a brute force method for identifying the bacterial communities, where we just, we extract the DNA from samples, and then we sequence the DNA, and it gives us basically what the bacterial community looks like. It does not give us information about whether they are active or whether they're alive or dead. It just gives us information about their presence or their absence. And so this is basically how they're going to be, how I'm going to be showing the results. And so you can then characterize, once you get the DNA, it identifies what kind of species we have all the way to species level, but sometimes it stops at order level, sometimes it stops at genus level, and then there's the most specific is the species. And so this is what it would look like for a red fox, but we're going to be looking at bacteria. So first we'll look at the bacterial orders. And so let me orient you to this graph. So what we have is 6 different samples. So from the Urinal-on-wheels, we have 3 samples where I added no acid, where I added a low amount of acid to control the urea hydrolysis, and where I added a high amount of acid to control urea hydrolysis. Then we have in the lab with the glass urinal, one with no acid in the trap. So I took a sample from that trap. I took a sample from the tank, and then I took a sample from an experiment where we added acid in the tank. And so this is the relative abundance of different bacterial orders. And as you can see, maybe it's harder to see, but the amount of bacterial orders for the urinal no acid and the lab tank no acid is a lot more diverse. We have a lot more orders than we do in the ones where we have acid added. So it looks like when you go from no acid to acid, you're increasing this big green bar, which corresponds to Enterobacterialis. And that you can see that as well here, where you go from the tank where you don't have a big green bar to a bigger green bar when it's added with acid. So then we move forward. So we're going down the scale from order. Now we're going to genus. And so these are the different samples that we have. We have the order, which I showed you before, and now we have the genus graphed. And so now it tells us closer to what is actually there. And so we can see that there are a lot of little bars here with no acid, as well as here in the tank with no acid. There's that bigger diversity that we saw in the orders. But then if we look at the experiments with acid, we can see that there is a selection for this big bar of Enterobacterialis. And inside the Enterobacterialis, no fecal contain, like fecally derived. And so then we went one step deeper. So this is the deepest step. We went down to species level. And this is the hardest level to get from sequencing DNA. It's very difficult to actually classify them. So there is this big chunk of the findings that were unclassified. So it's this big green bar at the bottom. But of the classified, the E. coli or Escherichia coli is the biggest chunk of what we found, which is like I said, it's from fecal contamination. And so the lowest amount of E. coli was found with the urine that was collected with no acid. And then he says once you add acid, if you go from low acid to high acid, you're increasing the relative abundance of E. coli in the stored urine or in the collective urine. And so what is the engineering significance? This is the first study to sequence DNA of bacterial communities in urine collected solely with non-water urinals. And the bacterial communities were influenced by the restroom ecosystem. We found that we did have some fecal contamination. And so this shows that it wasn't just from the interaction between the person using the fixture. It was also about what was in the air, what was being just walked in the air and landed into the urinal, and then the urine that was added on the urinal surface would then bring, I think, the fecal contamination into the tank. The results show that urine collected with non-water urinals can still contain a wide range of bacteria, including some pathogenic species, and implications on exposure. This has implications on the exposure to the maintenance staff that is handling the urine and the treatment technologies used for nutrient recovery. Especially if we're adding acid at the urinal and collecting the urinal as an acidified urine source. And so finally, we can't make assumptions about the bacterial communities when collecting urine with non-water urinals alone. And so with that, I'd like to thank you. And this is my contact information if you'd like to reach out.
Great, thank you very much. Very interesting. There's a couple of questions in here already. The first question is from Torre Nos. I hope I didn't mispronounce that. How much bacteria is present in urine that is not contaminated with feces?
I mean, we didn't achieve a urine that was not contaminated with feces. Even our lab setup was still, we still got some gut microbiome species that we identified. And so it's very difficult, but urine is not sterile. I mean, there's this big thinking that urine is sterile, but it is not. There is urine microbiome. And so the urine itself has its own microbiome that studies have shown. And then there's also the contamination that happens when urine is excreted and urine is collected. And so there's really just no way, I think, I mean, unless you go and do some medical ways of collecting urine where you're collecting it straight from the bladder, there's really no way of collecting urine without bacteria.
A question about that is whether this is a question from me. You call it fecal contamination. Is it established in your study that this is actually from feces entering the urine, or could it also be the biome of the urinary tract that is the source of those bacteria?
Yeah, so from the study, we identified 15 or 29 total genera, so genuses, and 15 of those, so 15 out of the 29 were from the urine metabolome. So like the microbiome of the urine was attributed to the 15, but 9 of them were from gut microbiome. So the gut bacteria, so that's like the fecal bacteria, E. coli, Klebsiella, all that was, so 9 of those 29 came from gut microbiome, and they are not found in the urine microbiome.
There's a bunch of questions here. We're not going to get to all of these, but I'll just pose one more to you, which is, how do the absolute abundances of fecal bacteria compare with hazardous exposure levels?
Yeah, so that's something that you can't study with the type of, you can't answer that question with the type of analysis that we made, because it just gives you the relative abundance. So the next step would be to do something like qPCR, where you know the amount of quantity of the bacteria that you're finding. And that's an important step, because what if my results show that there's a relatively high abundance, but that's just relative. What if there really is, it's very low and it doesn't affect public health? So that's the next step.
Great. Well, thank you very much.